Good evening, everybody, and welcome to you all joining us tonight. My name is Kay, and I'm part of the Musculoskeletal Health Australia team, and I would like to thank you for being part of our community webinar series. On behalf of Musculoskeletal Health Australia, we would like to acknowledge the traditional owners of the lands on which we're meeting today and extend our respect to all elders and any Aboriginal and Torres Strait Islander peoples who have joined us today. So a little bit of housekeeping before we get started. If you have any questions, please use the chat or the Q&A function to let us know what they are so that we can include them in our Q&A session. We would love to hear from you. And also, if you happen to have any concerns or questions, once the webinar presentation is finished, please feel free to contact us on our helpline. The number is 1-800-263-265, or you can email us at helpline@muscha.org. So tonight's webinar is on the role of the microbiome in a person's health, a subject that is of great interest in the health space today. The presentation will cover things such as the connection between the microbiome and your health, methodology that's been developed to evaluate the microbiome, and also talk about some of the studies in this area, as well as studies that are a bit more specific to what's known about the microbiome in inflammatory musculoskeletal disease. And we're very grateful to have Dr. Beverly Ng as our presenter this evening, who is a rheumatology consultant at the St. George and Westmead hospitals and has also completed rheumatology advanced training and a clinical research fellowship at Addenbrooke's Hospital in Cambridge in the UK on psoriatic arthritis. And she's also pursuing research that explores the microbiome in relation to treatment responses in patients with psoriatic arthritis as part of her PhD. So we thank you, Beverly, and we look forward to hearing from you. And on that note, over to you.
Thanks. Thank you, Kay, and thank you everyone for attending the webinar tonight. I will share my slides now. So I'll touch briefly on the microbiome and its role in health and disease, and followed by some more detailed studies in arthritis specifically. So without further ado, we all know that the microbiome consists of trillions of bacteria. We are specifically talking about bacteria, though there is a virome as well as a mycome, which encompasses viruses as well as fungal elements. But for our purposes of this talk, we are focusing on bacteria. There's an increasing interest in health and disease, and there's increasing technologies that allow us to analyze the microbiome, including 16S sequencing and metagenomics, which allows us to look more at what bacteria are in different parts of the body, and what functions they have as well. Some of the earliest applications for the microbiome include fecal transplantation for C. difficile infection. So it's a condition when we take too many antibiotics, and that kind of knocks out the good guys, so to speak, in the gut, and there's overgrowth of Clostridium, and hence transplantation with feces from healthy donors can help restore the ecosystem in the gastrointestinal system. So this diagram here just highlights the different pH levels in the gastrointestinal system, as well as different amounts of bacteria in these sections. This diagram highlights different organisms that are predominant in different areas of interest with microbiome research. And today we'll be mainly focusing on oral and gut microbiome. So what is the function of the microbiome really? It helps synthesize vitamins, amino acids, and some hormones. And it's very important in maintaining skin and mucosal barrier function, as well as regulation of our immune system. And I'll go further into that later in the talk. It also helps process dietary fibers that aren't traditionally absorbed by our body, and processes a lot of carbohydrates. This leads to production of certain gases as well as short-chain fatty acids. The key players here that a lot you'll see mentioned in the literature include butyrate, acetate, and propionate. Butyrate is actually really important as a fuel for colonic cells. So it's food for the cells in the colon and helps to maintain the gut barrier and suppresses inflammation in that area by maintaining a certain degree of oxygenation or low oxygen environment in the gut, which doesn't allow pathogenic bacteria, so bacteria that cause disease, to overgrow in the gut. So it's a really important player, as you'll see later on in a lot of studies. Acetate is the most produced short-chain fatty acid, and it's also helpful in the integrity of the lining of the gut, and is also used in cholesterol metabolism. Propionate is also involved in cholesterol metabolism. So here we have a better kind of diagram to highlight what happens at the gut level, at the lining of the gut. So you can see here, this is the colon and the lining of the colon, where all the bacteria are hanging out. And when there's butyrate, that helps to maintain low oxygen levels within this area, as I mentioned, which allows a certain type of anaerobic bacteria, so bacteria that grow in low oxygen environments, to survive and do their function there. And when this kind of ecosystem is in place, we call that a healthy gut barrier, where you have your normal function and reduction of inflammation. However, when you have any disruption of that environment and there's reduction in production of butyrate, that will trigger off what we call the leaky gut, or a pro-inflammatory state with the gut, which can then lead subsequently to different kinds of diseases. And there are different kind of situations where there's reduction in the butyrate producing organisms, such as use of medications or disease states where we observe this. This is a schematic diagram to look at the common bacteria we see in the gut. So again, we mentioned butyrate production. So firmicutes are, and especially F. prausnitzii, pardon my pronunciation, is a very avid butyrate producer. And so you will see in certain conditions, such as IBD or certain inflammatory arthritis, this is actually reduced. And interestingly, more recent studies, very small numbers, but in fibromyalgia, they've also noticed reduction in this organism. Then you have your proteobacteria, Bacteroidetes, as well as actinobacteria. And these all have their specific functions and vary in their kind of volume or composition, depending on disease or health states. So I'll just go a bit more briefly into how we analyze the microbiome in terms of the technology. So broadly speaking, there is 16S sequencing and metagenomics. So as a researcher, we have to collect the sample from the patient. So that could be a salivary swab or a stool sample. This needs to be temperature controlled, ideally at -4 degrees. Transported and stored. Subsequent to that, we have to extract the DNA through multiple steps in the lab. And once we isolate that DNA, we can then sequence it in a sequencing lab. This is through replication of one area on what we call the 16S rRNA gene, which is part of the bacteria. And we kind of amplify that region and see how many counts of different kinds of bacteria are present in that sample. And that allows us to kind of capture at that one moment in time where this sample is produced, the composition of the bacteria that are being grown there, And we can also estimate the downstream functional component from these bacteria. And that is through various libraries that have been developed over time on these bacteria and their functions. And by function, I mean, what do they do in terms of breakdown of carbohydrates or breakdown of medications, which is stored on various databases available to microbiome researchers. We also look at alpha diversity and beta diversity. So alpha diversity refers to the differences within an individual, and beta diversity is between individuals. So you'll see a lot of this mentioned in the research papers on microbiome. And you'll also notice that a lot of patients with disease or studies on diseased states highlight reduced diversity in disease populations. So without further ado, we'll talk about some factors that would influence the microbiome composition in the first place. So a lot of studies have been done on this, and they've highlighted a few key things. That includes diet, medications, physical activity, your geographic location, and importantly, from birth, that whole period of the birthing process, as well as breastfeeding, all impacts on our gut health and microbiome. And subsequently, different stages of life also yield different microbiome composition as well. So from birth, you can see here in this diagram that different methods of delivery would yield different composition of microbiome in infants. And subsequently, whether they're breastfed or formula fed also impacts the degree of, how much lactobacillus or bifidobacterium is in their gut. Relatively speaking, by the time you're 3, you start getting influenced by other factors, including your diet and your lifestyle as a child, and it stabilizes at that point. However, studies have shown that at older age, the microbiome tends to reduce in diversity, similar to what you have as a child, before you're exposed to all these different stimuli that you put into your gut. So it's rather interesting that at an older age, it does revert back to a lower diversity, closer to what you were as a child. So without further ado, I'll go further into some dietary compositions and how they impact the microbiome. There are a lot of studies on this, and I just summarized some of the major studies. So in terms of a very poor diet that is high in fat, high in sugar and protein, and low in fiber, this shows that there's reduced alpha diversity and reduced abundance of butyrate-producing species. As I mentioned previously, butyrate is very important in regulating the intestinal barrier, and reduction of butyrate can lead to pro-inflammatory state. Conversely, a Mediterranean diet, which is predominantly plant-based, minimally processed, with high, moderate fish intake, high fiber, and less red meats, there's increased alpha diversity in this group compared to, the relatively unhealthy diet we've previously mentioned. And interestingly, ketogenic diet, which is high fat and low carbohydrate, you observe reduced bifidobacterium, which is generally more prominent in populations that have higher carbohydrate intake. So, logically speaking, high fat and low carbohydrate in ketogenic, you get reduction in that group. There's also, with the production of ketone bodies from the ketogenic diet, you also have reduction in Th17, which is a type of T helper cell that can be pro-inflammatory. So there's reduction in that in the intestine. high starch and fiber diet can reduce pathogenic strains and increase microbial richness. So you always hear people say supporting a high fiber diet, and that is indicated in some studies that it helps with richness and production of the short chain fatty acids. Other types of mono and disaccharide carbohydrates also increase bifidobacteria, which is opposite of what you see in the ketogenic diet. And the low FODMAP diet, which is used in IBS, that reduces the overall bacterial counts, but the limited studies have not shown any change in diversity as yet. And it does reduce bifidobacteria. What about physical activity? So there are some select studies on rugby players who go on a really good diet and strict exercise regimen, and it's shown that they have greater diversity and greater production of short chain fatty acids in that group compared to sedentary adults or regular adults who go about their normal life. And so fitness and physical activity is associated with increased diversity. Interestingly, exercise that lasted longer actually showed the greatest change in diversity. And in a systematic review, which outlined these elements, they kind of highlighted different types of exercise. And there were most changes seen with aerobic exercise. And it can take anywhere up to 2 weeks for the changes to occur in the microbiome, with stability achieved at 6 to 8 weeks of persistent exercise. So here's the summary from that systematic review, which looked at the different kinds of exercise and what it did to the microbiome in that population. So interestingly, the low to moderate intensity activity was enough to increase alpha diversity in majority of the studies. And there was similar findings in the moderate to high intensity. But you can see here, even low to moderate intensity is helpful. Although there was not much change found in the HIITS, the high intensity interval training group or the sprint interval training group, we have to understand that these were very limited studies, only 4 studies in the HIIT group and 3 in the sprint training group. So I think replication of these studies would be very helpful to validate or, see if there's any change in the data from that. Now, what about medications? So yes, medications do change our microbiome. The most obvious one would be antibiotics, which does reduce gut microbiome diversity. So by knocking out the pathogenic organism that we want to target for whatever infection, it can knock out healthy gut bacteria and subsequently lead to what we call a dysbiotic state in the gut, and has downstream effects from that. Other less well-recognized medications that can alter the gut microbiome include very common medications such as proton pump inhibitors. So these are your Nexiums, your Somax, that are helpful for patients with gastritis, but it has been shown to lead to reductions in diversity and reduction in bacterial abundance, which can increase the proportion of the not so good bacteria, such as pathogenic E. coli. So even within certain bacterial groups, there are different strains, some that cause disease and some that don't. So it's quite important to recognize that can happen with some of these other medications, including proton pump inhibitors, as well as metformin and statins as well. There are many trials looking at supplementation with probiotics to enhance treatment response. So that's a converse. So instead of having a deleterious effect, there's a lot of researchers looking at how we may alter the gut microbiome with supplementation to see if that can enhance treatment response in different diseases. And this is still a work in progress. There are a lot of trials happening. So we don't have definitive answers as yet with regards to that. This is just a diagram to show you what happens when the barrier is disturbed. As we mentioned previously, if you have a medication such as antibiotics that increases facultative anaerobes by increasing oxygen levels after kind of reduction in bacteria that help that butyrate production, it becomes a disturbed space. Subsequent to that, you have, the new bacteria or pioneer species that try and come to help restore that healthy gut lining. So that's what you hope we will achieve, ideally, but that doesn't always happen. Now, what about the observations in healthy, like extremely healthy populations, so centenarians, right, globally? So this was an interesting kind of summary study in Frontiers in Microbiology, looking at various centenarians in different countries. And interestingly, there wasn't a unifying microbiome profile. And I think the most logical explanation would be that there are different dietary components in different countries. So, your Mediterranean diet more so in Europe, places like Sardinia in Italy, versus China and Korea, as well as Japan, where you have a larger proportion of patients, of people eating carbohydrate-based diets. So it's interesting to see that difference between all these countries. So there isn't a magic kind of microbiome signature for old age. So what does that mean? I think examining the function of these organisms should be explored further. And the common theme that the researchers found was that a lot of these people ate minimally processed plant-based diets, rich in complex carbohydrates, fruit, and vegetables. So that was a common theme amongst all the centenarians. And what a lot of the literature refers to as a healthy microbiome is a diverse microbiome with high richness, as well as a very stable core microbiota. So someone who has a very stable microbiome over time. And that really, as we discussed, it means that they haven't had many diseases, they haven't had to have a lot of medications to alter what's happening in their gut. And as we mentioned previously, in infants and elderly population, the microbiome is actually less diverse and increases after age 3 based on your lifestyle and diet and environment. What about the microbiome in disease? So as we mentioned, a lot of disease states, the microbiome has been observed to be in a dysbiotic state, or dysbiosis, as you can see here. And various amounts of diseases have been studied. These are just some of them. And more, this is being looked into to look for potential therapeutic targets and to look at how the disease came about in the first place. So there's a reduction in diversity in various disease states, and these are just some of them. I mentioned psoriatic arthritis there. And there's this state where they call low diversity dysbiosis. And it's thought to be driven partly by lifestyle factors, so diet, alcohol, as well as our medications such as antibiotics. And there's increase in facultative anaerobes and loss of butyrate producing bacteria. And this subsequently can lead to pro-inflammatory states and can be linked with diseases such as colorectal cancer, obesity, diabetes, as well as our inflammatory arthritis. And here we have a table where you can see which bacteria are more prominent in low diversity dysbiosis versus more healthy gut with butyrate producers. So your Rheumacoccus and Lechner-Spiraceae in the Clostridialis group. And what's the functional consequence of dysbiosis? So as we mentioned, you have your loss of your anti-inflammatory effects and the gut barrier is disturbed. So you have a greater chance of the bad bacteria or pathogenic bacteria hanging around in your colon and presenting as kind of immunogenic sources to your immune system. So, there's dendritic cells, there's cells in the gut that recognize some of these pathogens from that area, and that can trigger off a whole host of inflammatory responses. So again, a summary table of various diseases and the pathogens that could be linked to them and the subsequent inflammatory pathways that come from that. I won't go to too much detail with this because there's a bit more to cover. So again, another schematic diagram looking at different inputs into our gut system and what can happen as a result. So the green is meant to represent the good things, and the red is meant to represent the not so good things or things that alter it. Dietary fiber intake is definitely recommended for the production or for generation of short-chain fatty acids, as it promotes those bacteria that help with that. And also a whole host of other metabolic benefits, including improving insulin sensitivity, antioxidants, and improvements in lipid metabolism. Whereas your excessive sugar intake, too much protein consumption, or fatty acid intakes, as well as certain medications, can lead to the opposite. Now, what about arthritis? So we'll get into the heart of a lot of what I'm studying. So reactive arthritis, which is a type of arthritis that you get when you get a gastrointestinal infection or genitourinary infection. So often patients can get gastrointestinal infection, for example, with Shigella or Salmonella, and not soon after that, they may develop inflammation in their joints. That is a very clear proof of concept where a disturbance in the gastrointestinal system is leading to an inflammatory process that you can see clinically. And we have a lot of studies now on the microbiome in rheumatoid arthritis and other types of inflammatory arthritis, namely ankylosing spondylitis and psoriatic arthritis. And what we've noticed is that there is a different microbiome composition in patients with arthritis compared to healthy controls, much like the other diseases we've talked about. I just thought I'd include this. This is not part of the studies that I'm doing, but in gout, you can already see that there are differences in the microbiome in those with gout and without gout. And also those without symptomatic gout, so they may have high uric acid in their blood. They have a difference in the bacteria compared to people with active gout. So that's an interesting note. So people with active inflammation have a different profile of gut bacteria, and namely there's reduction in butyrate and propionate producing bacteria in the gout patients compared to those with just high uric acid but no gout symptoms. So moving on to our typical inflammatory arthritis. So rheumatoid arthritis, I'll talk about the studies on the oral microbiome. In the review that we did, there were 11 studies on the oral microbiome, and a lot of interest started in patients with periodontitis. There's a theory that stems way back that poor dental health and poor teeth dental hygiene can lead to microbial dysbiosis, so disruption of the microbiomes in the periodontal area. And this can then trigger off a pro-inflammatory cascade, leading to generation of the antibodies in rheumatoid arthritis and destruction of joints. That is kind of being studied here in terms of subgingival plaque samples. So dentists often can help us with that and get samples from subgingival plaque. More easier access way would be to look at saliva. So there are studies on both saliva as well as subgingival plaque, observing what bacteria are there. So a lot of the studies actually identified Prevotella in the oral microbiome for rheumatoid arthritis, especially new onset rheumatoid arthritis. So you'll see this come up a lot in a lot of the papers and a lot of the articles on microbiome in rheumatoid arthritis, that Prevotella is a potential trigger for rheumatoid arthritis because it's observed in a lot of medication or treatment naive patients who just who get RA in the early stages. So this has also been shown in mouse models where they have introduced Prevotella copri in particular to mice, and they've developed inflammatory arthritis, the mice who are infected with the Prevotella through a different vector. Okay, so in terms of other studies, there are limited longitudinal studies that follow patients over time, but in those specific studies, they showed higher levels of Lactobacillus salivarius. In terms of the stool microbiome, there are a lot more studies in rheumatoid arthritis compared to oral studies, and we included a total of 32 studies. Majority used 16S sequencing, and in this group, there was a bit more conflicting evidence on Prevotella copri. Again, it was more abundant in new onset rheumatoid, and there was reduction in the Prevotella with treatment. So with the methotrexate treatment, this organism reduced. And there were multiple studies showing higher abundance of specific genus, including Collinsella and Egerthella, which were associated with higher disease activity in rheumatoid arthritis, in addition to lactobacilli. Butyrate producing organisms were reduced in the treatment naive cohort. So that is in keeping with all the evidence from previous that butyrate is quite important. And there's a reduction in Faecalibacterium, which is one of the butyrate producing organisms. What about prediction of treatment response? So interestingly, the presence of Prevotella helped demonstrate that those patients who had that in the beginning had a better response to the treatment. And the ones that did not respond to treatment, and we're talking here about methotrexate in most of these studies, had abundance of Lachnospiraceae and Adela crutia than those that responded. So it's interesting that we can observe some of these. Of course, these are very small studies, so we would need to revalidate this in larger cohorts to see if we can reproduce that. And in a lot of the studies nowadays, you can see that they try to integrate some of this information, like the bacterial information, with evidence from their blood samples, so genomic factors from their blood tests, as well as other patient factors to try and predict it using an AI model or machine learning model to predict whether they respond to a certain treatment or not. Now I'll focus on one specific study that looks at this from New York and Spain. And one of the major players on inflammatory arthritis and the microbiome is Jose Scher from New York University. And they looked at whether the pre-treatment gut microbiome is associated with lack of response to methotrexate. So I'm looking at something similar in psoriatic arthritis. So we were trying to see patients with rheumatoid arthritis without having started any therapy, no methotrexate, no medications yet. And we look at what's happening with their gut bacteria. And can that help to see whether they will respond or not to methotrexate? And so they, in terms of their results, they divided kind of the patients into methotrexate responders and methotrexate non-responders, and they found some significant differences there. And here I won't go into too much detail, but basically there are certain metabolic pathways or functional pathways that they found that are associated with no response to methotrexate or poor response. So a lot of these pathways are to do with methotrexate metabolism and folate metabolism, which makes sense, as you know, that's the drug of interest. And then they inserted all these type of functional pathways into a prediction model, and selected a few, and that created the highest type of accurate prediction for patients who may not respond to methotrexate. And they validated this in another cohort of patients, and that allowed 80% of patients to be correctly classified. So that's quite impressive, that this kind of model can help predict who can or cannot, who will or will not respond to the methotrexate. So I don't, for the patients in the audience who have rheumatoid arthritis, I guess oftentimes in the clinic, we would go through different kinds of medication, and it's hard for us to know whether that will help you or not. So this is actually a step forward in terms of what we call precision medicine, or being more accurate and personalized, by looking at whether the bacteria can help us predict better which medication you may or may not respond to. Of course, this is still in very preliminary research stages, but it is somewhere that we definitely need a lot of more research on to help reduce unnecessary steps in terms of medications. And in terms of their discussion in this paper, you know, by using their model, 5 out of 6 of the new onset rheumatoid patients would get different therapies based on their prediction model. So that may help those patients to kind of use a different medication faster and avoid all the problems that they had, or the poor response to the methotrexate. Obviously, there are caveats to the study. There's limited numbers, and you know, where it's good faith that the patient is taking the methotrexate accurately, and there are no other things influencing the methotrexate pathway at the time. But this is kind of a first step into looking at how this may be of use or helpful in a clinic setting. So that was a bit of a segue, but going back to our other types of arthritis, so ankylosing spondylitis, there were 28 studies, 19 with 16S sequencing and 6 with metagenomic sequencing. And there are some longitudinal studies examining the effect of treatment, including biologics. And they identified distinct bacterial signatures that predict treatment response. So that includes things like Burkholderia and Dialister species, which correlated with good response, albeit the follow-up was quite short. I think they did 3 months and 6 months studies. So we are trying to see if there are more studies that come out with longer follow-up to see if this is sustained. And there are also species identified related to disease activity. So looking at their BASDI scores, there's higher degrees of Rhuminococcus navis in those with higher disease activity. And interestingly, as we mentioned, there is depletion of butyrate producers in those with active disease. So specifically Faecalibacterium prausnitzii, this has come up a lot. And the population is restored after treatment. So it shows that with these longitudinal studies, there is kind of going back to a more normal or a more healthy gut lining with these better or bacteria that produce butyrate after treatment. What about psoriatic arthritis? So this is actually an area of need. Currently, there are very few studies looking at the microbiome in psoriatic arthritis, even though it is still on the spectrum of kind of axial spondyloarthropathies and, you know, IBD, that spectrum of disease, but specifically looking at a purely psoriatic arthritis population, that's still a underserved community in the microbiome world. Interestingly, in 2020, a Danish study group tried to look at fecal transplant in psoriatic arthritis group. So they looked at fecal transplantation in psoriatic arthritis compared to a sham fecal transplant. And unfortunately, that was a negative study. It didn't show any better outcome in those who got the real fecal transplant at 26 weeks of follow-up. However, there were no serious adverse events in both groups. And it was a very small study, so 15 patients in each group, and very specific kind of phenotype or type of psoriatic arthritis that they studied. So for those who know about psoriatic arthritis, there are very different types of psoriatic arthritis, including those with just a few joints involved, or those with predominantly skin and nail disease, or spinal disease, or those with dactylitis, like swollen fingers, swollen digits, or enthesitis, which is more Achilles tendon problems. So it didn't really capture all of those different groups. So I think it's a watch this space type exercise. Interestingly, a report came out quite a few years ago now from Adelaide, where they did a fecal transplant on a patient with psoriatic arthritis. And not because of the arthritis, but for Clostridium difficile infection, which is a standard therapy where fecal transplant is used. And her psoriatic arthritis was actually very difficult to control, and it wasn't controlled at the time when she had this Clostridium difficile infection. But after the fecal transplantation, she actually got better, not just from a gut perspective, but also her arthritis. And this was sustained for at least 1 to 2 years. And she didn't need to go back on her DMARDs at that time. However, that's just a single study, but I found that quite interesting in that the restoration of her gut bacteria did help in some way with the inflammatory component of her joint pains. So there may be something there, but more studies need to be done. Okay, so some summary of the findings from the inflammatory arthritis studies. Higher disease activity was reflected in the gut microbiome in these patients where there's lower diversity. And the bacteria from particular genuses, so including Prevotella, Collencella, and Lactobacilli, are implicated in triggering or being pathogenic in the development of rheumatoid arthritis, as they're observed more in new onset rheumatoid arthritis without any previous treatment. And mouse models show development of disease when particular species are introduced, including Prevotella copri. And researchers have looked at Prevotella copri quite closely. And there's a great deal of genetic variability in this species itself, where there are different strains. So there are some strains specifically that cause disease, and this is replicated in those mouse models. And we also know from some of these studies that there is a distinct functional profile, so those metabolic pathways and types of bacteria in those who do respond and those who don't respond to treatments such as methotrexate in rheumatoid arthritis. One common theme we see is that there is depletion of butyrate producers in the rheumatoid arthritis and ankylosing spondylitis groups, which is consistent with a lot of the other disease states we have observed and discussed previously in our talk. There are limited studies in psoriatic arthritis, so this is an area that really needs more input and research. So what are some potential directions in terms of the microbiome in this area? So looking at interventions, so fecal transplantation. However, we have our barriers, including patient acceptability and selection criteria, and where are we getting our donations from? Secondly, probiotics. Actually, in this space, there are ongoing clinical trials in the rheumatoid arthritis population. So if you are attached to a hospital, I know Westmead is currently conducting that with the rheumatoid arthritis patients. It is still a kind of experimental phase. So looking at whether supplementation helps with a treatment response. And there needs to be a lot of replication of these studies to really observe a good effect. The main direction that we can see in this space in terms of testing, so just doing testing on patients, so gastrointestinal stool testing or ORSIT testing, is how this may help fit into a model to predict treatment responses or predict whether you will develop a certain phenotype or characterize the disease state. So that's kind of where I feel the microbiome will be helpful in a big puzzle. So it's one piece of the puzzle, I think. There would be integration with other factors such as gene testing, and shifting the focus away from just looking at which types of bacteria or the composition of your gut bacteria, but also looking at functional analysis. So the downstream metabolic effects. So that concludes my talk for now. I'd like to acknowledge a few people. I'd like to thank Musculoskeletal Health Australia for the invitation, Professor Mercer Lasser, who is my supervisor for my PhD, Professor Georgina Hold, who is also a microbiologist at the Microbiome Research Centre at University of New South Wales, and all the other staff there, as well as Arthritis Australia for funding some of my research on psoriatic arthritis and the microbiome. So thank you for that. I think now we'll go into our question session.
Yes, we will. And yeah, thank you from us as well, Beverly. That was very in-depth, great information, and providing some hope. And I think you can see the emojis on the screen coming up that people really appreciated your presentation. So yeah, thank you. And thank you for the people also who've sent in some questions. We've got a little bit of time, so we'll try and get through all of those. Lorna made just the comment, which I think is common to many people, that going through that period of trialing medications is really almost the worst part. So very hopeful about the research that you've been talking about. But she did wonder whether the testing the microbiome in future could be used in her case, where she's seronegative and has no psoriasis, but does have inflammatory arthritis.
Yes. So actually that is a very good question. It is actually one of the kind of study priorities we were trying to include in one of our studies, trying to look at characterizing seronegative patients who have been classified as rheumatoids versus those with psoriatic arthritis. So it's hard to do that without a consistent definition. I think you can see in a lot of the current studies on rheumatoid arthritis, they'll give you a percentage of those who are positive, like seropositive, but they don't go further into, oh, was there a difference in the seropositive group versus those who weren't? They may just say that in the seropositive group, there was more of this bacteria, but I feel that this is an area that needs to be worked on, and it will help with, diagnosing things in the future if that's used as a common testing panel, because there is no blood test, and the stool may be a biomarker for that. So I think that's a very good question, and if there are other researchers or other people with more funding, they should look into that.
Great, thank you. Chris asked if rheumatologists can order testing of the biome before starting medications early in the diagnosis, or is that still in trials phase?
Yeah, so I'll answer that question. Currently, rheumatologists don't routinely order that because it's not funded by Medicare or PBS. It would purely be in a research study setting. So there are certain centers that are doing that depending where you live. I think in New South Wales, in Sydney, North Shore they have the largest data bank for that. They've applied for what we call a biobank. So a lot of the arthritis patients who go through there or go through some of the trials there would have different biospecimens collected, including blood, including stool. So but in terms of a wider population, that's not available yet. So if you're interested, you may want to look up in your area. There are different centers that do it, particularly Queensland. I know there's certain researchers who do it up there, who may offer that kind of testing, but it may not be kind of available to you like what the commercial microbiome testing centers do, where they give you a breakdown of all the bugs, because it will all be de-identified and kind of used in a pool of analysis with big population. Yeah.
Yeah. Okay. Thank you. Julian is asking how much research has been done, or has there been any research done on treating the microbiome related issues by changing diet as opposed to medication? Have you got any information on that?
Yeah, so there was a lot of interest in this, and I think it was mainly looking at Mediterranean diet as an intervention for rheumatoid, and there were some positive effects in that group, but I don't think they looked at the microbiome specifically. I think it was like a previous intervention study of Mediterranean diet in RA. I think this is something that really needs to be looked into. And actually, a lot of the studies neglect to address that issue about the diet in the study itself. They kind of just look at, oh, this is rheumatoid and this is what was in their gut. It didn't examine what the composition of their diet was. So I think that is something that, should be looked into and would be a research topic.
Okay, great. Thank you. Denise is asking about biologics. So you talked a lot about methotrexate being used in some research quite extensively, but any other medications or biologics, have they been used?
Yes. So there have been some studies on TNF inhibitors as well as IL-17 inhibitors. But again, these were fair and far beyond. So 1 or 2 studies in each group, so rheumatoid and AS. So it's hard to draw any conclusions, but they did both identify, kind of restoration of the microbiome back to a more healthy microbiome after the treatment. So yeah, so there is a space to watch. And actually, there are some trials looking at kind of weaning of biologics, and then we'll be collecting biospecimens in those groups to see what happens.
Okay. I might combine, because there seems to be a sort of common theme here with a few questions, just your thoughts on taking any particular probiotics. And also you talked a lot about diet and comparing the different types. So whether there is a particular diet or nutrition plan that perhaps can help people with RA or other inflammatory disease.
Yeah, so I think that's a very common question, and thank you for asking that. I think I tried to go through the different types of diets and how they can alter the microbiome. But again, it's a very individualized thing. It's a very case by case thing. Currently, there are some studies on Mediterranean diet that show some positive effects in RA patients. But obviously that may not be applicable to everyone, or it may not be the preference of every patient. You also have to look at risk factors for yourself. So let's say you have a high risk of diabetes or a high risk of hyperlipidemia or cardiovascular disease, and you may want to modify your diet according to that as well. So I think what's more important is something that you can maintain and not develop those kinds of diseases like diabetic and cardiovascular problems. There hasn't been much to show, specifically what probiotic is good in rheumatoid arthritis simply because of the lack of studies. But I think this trial will help to answer, the one on the probiotics may help to answer some of those questions.
Okay, thank you. And I guess has there been any indications in some of those studies about foods that people maybe should avoid if they have arthritis or other inflammatory diseases?
Yeah, so I think a common theme is basically like a lot of highly processed, high sugar, high carbohydrate, like processed carbohydrate foods are not good for inflammation in general because of the depletion in the butyrate producing organisms, as you can observe in obese patients as well as diabetic patients. That group has a kind of a similar profile in kind of pro-inflammatory process. So I think I would avoid those types of diets with high process carbohydrates. And I hope that answers the question. But from what the centenarian studies show, I think it clearly shows that those who live longer are those eating plant-based or fish diets with very minimal processed foods. So I think we follow that. You can't go wrong.
Yeah, sounds good. Jill has just wondering, perhaps you might be able to answer this within your network.
Yes.
Is there anybody doing research and treatment in Perth or Western Australia in this area, especially for fibromyalgia?
Not that I'm aware of, but I can we can certainly if you live in Perth, you can certainly contact your rheumatologist and see if there's someone who is interested in doing that.
Okay, great, thank you. And I guess there's obviously a lot of research that's happening at the moment and a lot of interest from consumers, patients. So have you got any resources or sites where people can keep track or be informed reliably and evidence-based that they can use to help inform their decisions?
Yeah, so I think kind of randomized control trials would be the most reliable in terms of intervention. So things like probiotics, fecal transplant, I would that's the highest grade of evidence you can use. In terms of general knowledge, there are some books out there that you can read. So some from David Mosley, I think he has a whole series of gut books, but that's just more, and that's more for a general population where he explains different kinds of bugs. But that by no means is translatable to, what you need to take or how you should, what supplements you should have. So it's just a more general knowledge type thing.
Yeah.
But some of the reputable journals, any peer-reviewed journals, so anything from the gut to arthritis, the arthritis journals, um, those would be good ones to look at and randomized controlled trials. And you can always ask your rheumatologist about their opinion. But as you can see, it's quite a young field, but rapidly developing. So there's a lot of new
Yes.
things coming out. Yeah, so I think that's quite good. Getting the microbiome tested, I think that can be expensive, and there may not be a consistent kind of lab currently that's validated. So, and your rheumatologist may not be familiar with what that is saying. So, it may, you can do it for your own interest, but as I mentioned, there are a lot of things that can alter it. So it depends on when you're having it done, what medications you're taking at the time. So you have to kind of interpret it with caution at the time.
Yeah, that sounds great. Thank you. Annette has had the question, which perhaps other people are wondering about, whether this information and research, because we've talked a lot about RA, a little bit about psoriatic arthritis and other disease, but whether that could be transferred across to things like OA or other arthritis diseases.
Yeah, so I think there are some studies on osteoarthritis, not as many as the inflammatory arthritis. And for the psoriatic arthritis, we're developing that. So I've done some research on that, and there are some other groups in UK and kind of worldwide that are looking at that group specifically, just because the disease is so different, even amongst itself. There's so many different phenotypes.
I'm just having a look to see if there's any last questions before we um, finish up.
Yeah, so I think
Um,
cost. I think Denise asked about the cost. Is it expensive to test the stool? So yeah, when I was doing my research, it was quite expensive. That's a rate limiting factor. But I think over time, if this is getting more common and there's more applications, potentially it can be reduced. So let's say we're testing 80 samples of poo, like separately, where the DNA is extracted, it would be anywhere from $4,000 to $5,000 for 16S. sequencing. But if we're doing metagenomics, which tests the whole genome, that's a lot more expensive. It can go up to $10,000 for 80 samples, for example.
Wow, that's yeah, quite a cost.
Yeah. So I think
Oh,
metagenomics is kind of still far more for research, but
yes.
6S may become more accessible over time when the technology gets older and more people use it for different
Yeah.
studies. Yeah.
Yeah. And Beth has a question which might sort of carry on from the gene or stool testing, is whether things like leaky gut could be visualized, say during a colonoscopy. Is that possible?
Yeah, so I'm not a gastroenterologist, but I think they can do biopsies, which can show, potential inflammation in the gut. But it's a bit difficult to, I think, macroscopically look at it when you're in doing a scope test. But I think they can sometimes see if it's inflamed during the when they take a sample and look at it under the microscope, or if they measure certain chemicals like TH17 or certain, chemokines at that site, to see if it's inflamed. Yeah.
Okay, great. Well, we've just gotten to 8 o'clock, so we've run out of time. But so just wanted to say thank you again to everybody who tuned in, and thank you for your questions. Apologies that we weren't able to get through them all, but we really appreciate your interest. And if we can, we'll work with Beverly maybe to answer a few more of those offline, and just include them when we send out the recording. And again, thank you, Beverly,
No
for
worries.
coming along tonight. We really appreciate it. It was a great presentation. I think we've all learned a lot. So yeah, thank you so much.
No worries.
Thank you to everyone. Have a lovely evening, and we hope to see you at the next one. Take care.
Thank you. Thank you, everyone.